Food is such an important and integral part of our lives. It provides us with nutrition and energy to lead a long and healthy life. However, consuming food in high amounts, or if it is unsafe, can lead to more than 200 different types of diseases, which are called foodborne illnesses. About 600 million people, so almost 1 in 10 people, globally fall ill after eating contaminated food. About 420,000 people die each year worldwide. Evidently, these foodborne illnesses present a major concern, so let's examine their development. Foodborne illnesses are infectious and toxic diseases which are caused by contaminants including bacteria, viruses, poisons which are also called toxins, parasites that can live in the intestine, mold that makes toxins, and bacteria that can carry or make toxins. There are more than 31 foodborne agents. Many of these foodborne illnesses, such as those caused due to toxic chemical contaminants, may possibly lead to long-term health issues such as cancer. A lot of the time, people use the term food poisoning to describe all foodborne illnesses, which is not wrong. However, your healthcare professional may use more specific terms. Foodborne illness includes all illnesses from any contaminated food or beverage. On the other hand, food poisoning is a type of foodborne illness caused by a toxin contaminating food. So what are some common symptoms of foodborne illnesses? Some common symptoms would include a headache, upset stomach, diarrhea with bloody spots, vomiting, stomach pain and cramps, or even a fever. Foodborne illnesses can originate from many different sources. The problem can begin during any point of the process of growing, harvesting or catching, processing, storing, shipping, or even preparing food. This can be due to things like poor hand washing, lack of disinfection, or improper storage. Bacteria that cause these foodborne illnesses can also spread from bodies of water or from exposure to animals carrying the illness. For example, E.coli infection caused by Escherichia coli bacterium. The timing of symptoms due to E. coli contamination is usually 3 to 4 days and possibly even 1 to 10 days. The most common source is raw or undercooked meat, but it can also be due to unpasteurized milk or juice, soft cheeses from unpasteurized milk, and fresh fruits and vegetables. To deal with this, a team of researchers at McMaster University, led by Dr. Tohid Didar, has come up with an easy way to determine if food, specifically meat, is contaminated with potentially deadly E. coli bacteria. So the technology which the team has come up with is called Sentinel Wrap, a tiny transparent flexible sensor patch. Using an automated inkjet printer, it is printed with harmless DNA molecules. When in contact with the food, it can signal food contamination in real time. The patch can be incorporated directly into food packaging, where it is in touch with the food and can monitor the contents for E. coli. The patch has no effect on the contents of the food. This technology can be a breakthrough, since it has the potential to replace the traditional food packaging labels, such as best before dates.
In an interview conducted by CBC News, Dr. Didar points out that the issue with current food packaging is that if we want to find out if there is any bacteria present in this packaged food, we need to open the package, take it out, take it to a lab, and then process it.
This is also a very resource and time-consuming process. This patch can tell us whether meat and other foods are safe to eat or need to be thrown out without the need to open the package or manipulate the sample. But how does Sentinel Wrap work? The patch is a sensing surface that generates a fluorescent signal in the presence of specific target bacterium. Sentinel Wrap is coated on one side with small droplets of DNA molecules known as DNA zymes. A DNA zyme is a piece of DNA where the sequence has been selected such that it has a specific activity in the presence of a specific target. In this case, the sequence is designed so that if E. coli is present and comes into contact with it, that DNA is going to be broken in a particular location, which creates a fluorescent signal. Users could read that fluorescence to determine if the food inside the wrap was spoiled by using UV light or a simple app on a smartphone. This is very convenient. As Dr. Tohid Didar stated in a McMaster Engineering highlight video,
when the bacteria are present, if you put it in the packaging, you can actually see the presence of those dots, that they shine up, compared to the samples that do not have the bacteria present in them. And then you can detect the presence of the target bacteria.
To make this a bit simpler to understand, we will use an analogy. The DNA zyme is like a rope, and there are scissors that are going to cut that rope in a specific place. Those scissors are the presence of E. coli. On one end of the rope, you have a fluorescence molecule. On the other side, you have a quencher. If the rope is intact, they cancel each other out. But if you cut it with the scissors and the two pieces become separate, now the fluorescence can be detected because the quencher is no longer attached. You may be wondering, what are the benefits of using this technology over current food packaging labels? First off, it is easy and cheap to manufacture. The patch is easy to manufacture as the DNA molecules used to detect contaminants can be printed directly onto the patch. The patch is printed onto plastic wraps and uses extremely cost-effective DNA molecules in tiny amounts. All of this minimizes the cost of production for the patch to only being in the range of cents and not dollars. Another benefit of this technology is that it has simple implementation into current food packaging plants. Given the simplicity of manufacturing the patch can be easily incorporated into the production line of food packaging plants. It will also help reduce food waste. By using the patch, we would be able to definitively tell if the food is contaminated or not, thus allowing the user to know if the food is indeed spoiled before throwing it out. It can be used in the store but also at home. Carlos Felipe, another member of the research team, stated in a McMaster Engineering highlight video that this patch will be ideal for supermarkets to determine if stocks of food are safe for consumers to buy or need to be thrown out. Another advantage of using this technology is that it can control foodborne illness outbreaks. By monitoring for pathogens such as E.coli in food packaging, these outbreaks can be stopped or minimized. And last but not least, it can also help reduce severe foodborne illnesses and related deaths by preventing the consumption of harmful contaminants. So, what is the future of Sentinel Wrap? The research team is working on developing a multiplex system where multiple bacteria and pathogens, other than E. coli, can be detected by the patch. As of 2018, the team hoped to partner with industry partners and regulating agencies to take this invention to the next step, where it can be incorporated into food production and distribution. The technology used to make the patch can be applied to other pathogen-finding purposes as well. For example, this technology can be used in bandages to tell if a wound is infected or not, or even in medical equipment or tools to ensure that they are sterile prior to use. There are many applications and uses of this technology. It is now up to research to determine what path it can take, but most certainly it has a huge potential in helping alleviate the burden of foodborne illnesses by being implemented into current commercial food packaging labels.